Okay, I think I'm going to get started now. Welcome, everybody. It's a special treat for me to introduce Larry Abbott. Larry is one of three well-known Larrys who are neuroscientists who attended Oberlin College. They range from A to Z. There's Larry Abbott, Larry Squire, and Larry Zipursky. This Larry majored in physics, but was too restless to stick around for graduation, so he left and contemplated his next move, which eventually turned out to be to return to physics. He got his PhD at Brandeis, working in theoretical particle physics with Mark Schnitzer's father. He took a position in theoretical physics at SLAC and worked at CERN. Also, those are probably places that have about the same ratio of experimentalists to theorists as we have in neuroscience. He came back to Brandeis as a faculty member in physics, and he did theoretical particle physics for a number of years and was tenured in the physics department, and then made a transition to neuroscience, and in fact switched affiliation to the biology department. He left Brandeis for Columbia 15 years ago or so, and has gone on to win probably every award or prize that's available in theoretical neuroscience. Probably to neuroscientists, which is to say experimentalists, he's best known for his work modeling experimental data, and that's for good reason. He has an ability to talk to experimentalists, and he has a taste for the concrete details of experiments, and he's had enormous success in that area. He's also had tremendous impact in theoretical neuroscience, computational neuroscience. He has done work on problems like the stability of toy model neural networks, various aspects of memory. He's worked on deep feed-forward neural networks and on training recurrent neural networks. And I think that in one of the couple of previous seminars, he spoke about training recurrent neural networks. And it was a talk filled with animations of eigenvalues and things like that. I think that this time the talk is going to be more on the making contact with experimental data, although I think I detected in the abstract some hint of implications that are broader than that. In any case, whatever it is that he's going to talk about, I'm sure it's going to be a lot of fun. So please join me in welcoming Larry Abbott.
Thank you, Kenny. It's great to be here to see so many friends, Kenny and many others that I'll enjoy spending the day with. This is the star of the show. It's called the elephant nose fish, although this is a lip. It's not actually a nose. And it's an electric fish, and I'll get into it in a second, but Kenny kind of got me right because I'm immediately going to go and talk about a little bit more theoretical problems. So what's the theme here? I think in order to get through all the detail, and there's a fair amount of detail here, it will be useful to try to keep track of what the theme is. And the theme is learning in layered networks. So, of course, plasticity and its effect, you know, the effect of changing these synapses on a neuron has been studied enormously in neuroscience. But it's almost always at this level of saying, well, you know, you can do various manipulations, that synapse will change, and it will change the response of this neuron in a certain way. And if you go to a kind of machine learning context, often there's an error signal associated with this output. You want the output to do a certain task. And the error signal is used then to control this plasticity. And that's the easy problem in machine learning. That's the solution to this has been known for many, many years. So that's mostly what's been studied in biology, and it's the easy part in machine learning. Things get a lot tougher if you add another layer into the problem and you look at these black neurons that I've drawn and how they feed into the system. And in particular, you start thinking about how should you change a synapse at this layer in order to improve that error. This is because, of course, that error is an indirect function of what's going on back here. And, you know, I'm sure you all know that back propagation of errors is the machine learning solution to this. I think it's generally believed that's not what's going on in neuroscience. So there's a real question of how the many layers in our brain, not directly associated with an output, know what to do, how to change their response in order to make things better, make things work better. So basically what you need here is an error associated with this neuron in order to know, for example, how to change that synapse. Okay, so I'm going to explore, because this is what the electric fish does, the simplest thing you could think of here. And that is basically, I guess the theme is, you know, everybody do their best. So the idea is that you apply the same error to everybody. And learn on that basis. This is something that I'm sure didn't originate with David Cicillo and me, but we did it. Lately, there's a group at DeepMind looking at this kind of learning. And so, since it's important to understand, let me just give an example. So imagine I had a picture here, and I tore it up into little bits, and I gave everybody in the room a little piece of the picture. And I could say, well, our collective job is to figure out what's in the picture. So the cost function there would be approximate the true, you know, object in the picture as closely as possible. And if I gave that as a task to everybody in the room, then, you know, somebody might yell out it's a car, somebody might say it's a tree, somebody might say it's a horse. And, you know, you're all doing your best, but it's probably not the best way for us to collectively solve the problem. So the problem here is that the signal that minimizes an error is often not the best signal to transmit to the collective. So we'd do a lot better if somebody would say, oh, I know there's an eye in my picture. I don't know what the rest is, but I see an eye. And somebody else says, I see a tail. And, you know, we'd pretty quickly come across the solution. So the basic problem in this kind of learning is this problem, that what you transmit is not necessarily, shouldn't necessarily be your best approximation of the final answer. It should be something that you determine would be useful, for example, at the output. And the great thing about backprop is it figures out what that is. So I'm going to talk about a system. Let me just say one more thing here. Of course, the advantage of doing this is there's just a global error signal. It goes to every neuron in the brain, and every neuron does the same thing. So there's something to be said about a learning scheme where everybody just tries their best. But the disadvantage is, again, this problem that a poor approximation of the truth is probably not the best signal to send downstream to help the collective problem. And so I'm going to talk about how the fish resolves exactly this problem. You'll see the fish has this same problem. The fish actually uses this global error type scheme, and it runs into this problem, and it solves it. All right, so that's where we're getting today. All right, so back to the fish. So this is an electric fish, and it's electric in two senses, and it's important to keep those two senses straight. So first of all, like many fish, catfish, sharks have this. It has on the surface of its skin many what are called passive electroreceptors. This is not what makes an electric fish, but it has these. And those are extremely sensitive to very small electric fields, and so they can sense the movement of prey in the water, things like bugs. When the bugs flex their muscles, you get an electric signal, can sense those. Sharks use these for example, finding prey that are buried in mud or something like that. So it uses this system. This is called the passive system, and it's the system I'm going to talk about. The problem in the electric fish, though, is that it also has an active sensing of electric fields, and it generates its own electric field. It does that from a modified muscle in the tail that produces a very sharp shock in the water that is much, much larger than what the prey uses. Now, this system is used also to find prey. It's also used for communication, but I'm not going to talk about it at all except as a source of interference for this system, because the circuit I'm going to talk about is the circuit designed to remove this source of interference from the system. Okay, so anyway, when the tail shock goes off, it generates this huge field, and this is actually a recording done in Nate Sautel's lab, where you can see that these are the times when the fish, these red lines, made an electric organ discharge. You'll see EOD on my slides. That's electric organ discharge. Generated a large field in the water, and that caused this huge response in one of these electroreceptors. On the other hand, in this experiment, and I'll come back to this experiment at the end of the talk, Armin, who did the experiment, also simulated a prey, which is these small wiggles. And you can see that the prey, in this case, even though this prey is probably anomalously large, is swamped by this large signal from the self-response. So as I mentioned, this circuit is designed to get rid of that background, the noise that's caused by your own system, and to find the prey. Okay, let me see. So this system has been studied in Kurt Bell's lab in a really beautiful series of papers that I recommend to you very highly. And also very beautiful modeling by Patrick Roberts. So this is as elegant as both theoretical and experimental neuroscience get. Beautiful stuff. But my work is done with these people. Solomon Mueller is a graduate student working with both me and Nate, and has really done all the modeling stuff. Nate did a lot of the experiments himself, and Abby also joined in some of the experiments. So that's the team, and that's the fish. Okay, so let me now go through this in more detail. So there's a command nucleus in the basal part of the brain of the fish that sends a command back to this organ, electric organ, and causes the shock. So that's one part of the circuit. There's this circuit called the ELL, electrosensory lateral lobe, that receives electrosensory input from all over the skin and processes it. And the key to the whole system is that there's a corollary discharge signal that goes to the ELL. So the ELL gets a signal every time the fish decides to send a shock in the water. And that's the basis of this system. The fish knows that it discharged its electric organ, and what you'll see is the ELL is going to generate a prediction for the fish's own electric field and remove it from the sensory stream, thereby revealing that bug. Okay, so just to mention that this system is very much like systems that we have, that we use all the time. We may not realize it. Correct. And when I walk around, I know that it's me moving, not you guys moving, because I have the motor signals within me. So we do this all the time. This is just kind of an extreme example and a very nice example to study. All right, so now let's go into the ELL, and I'll take you through the circuitry and set up the problem. So here's the command nucleus. It's going to generate this electric organ discharge signal. Maybe I'll point over here, then I can face you a little bit more. That generates a shock in the water. That shock in the water generates a response in the electrosensor sensors. That's the big spike that you saw, a spike-like thing that you saw. But in the water, there might be a bug generating a much smaller fluctuation in the electric field. So I've represented that here. And of course, what comes into the sensory system is the sum of these two. And the fish's problem is to find these little wiggles on top of these big wiggles. By the way, these electric organ discharges happen at a few hertz up to tens of hertz. So it's not like the fish can wait between pulses and find the prey, because these are going to be wall to wall. I only showed you one, but there'll be another discharge right then. They're just going on continually, so you can't. Yeah.
So these bugs are also at the surface typically? Are they generating capillary wave as well as electrical signals?
Yeah, that's interesting. Probably, but they can definitely find things in the water submerged, because people, play with them that way. Sometimes they may also use their sensory lateral line lobe system to detect them. Yeah, you're right. Okay, so anyway, afferents bring this into the ELL. There are what are called output cells, and through an interneuron, those output cells are driven by the sensory response. And if this was the whole story, this signal would come in, and it would more or less come out through the output cells, and we wouldn't have accomplished anything. This would then just be a relay. The key is, though, that as I mentioned, there's a corollary discharge and a set of granule cells that form parallel fibers that send into this system. The idea is a command has been generated, get ready, because there's going to be a strong electric pulse entering through the sensory pathway. And that is conveyed to these cells through this parallel fiber system. One of the things that we did with Nate's lab before was to look at the responses of these granule cells and realize that they form kind of an interesting temporal basis. Although this is a very short pulse, that pulse sets off a whole time sequence of activity in the granule cells. And so from this basis, this output cell can, through these synapses, generate an estimate of what it thinks the sensory response is going to be, what it thinks that will be. And in fact, that does happen. There's plasticity at these synapses, and what's called a negative image is formed. So there's an inverse pattern here, inverse to this, and it used to be that when always in Kurt Bell's talks and the talks, I've given talks before about this, that this was kind of the whole story. The argument was that this cancels, this thing, leaving a flat output. That is not true, and I think everybody knew it's probably not true, because it leaves out the bulk of the cells in the system. This is a common thing in neuroscience where you have the answer, only you haven't explained 90% of the circuit. And the remaining part of the circuit are what are called medium ganglia, or MG cells. They're both the majority of the cells, and their synapses up here are 90% of the granule cell synapses. So this is the heavy learning machinery here, and in fact, the learning had been studied in this system, but the cancellation had been studied in this system. And I'll give you the answer that I'm going to explain to you during the talk. Well, let me just do this first. So here you can see how many more MG cells there are than these output cells. And so, as I said, the majority of the system had not been even described. By the way, this language you might recognize, granule cells, parallel fibers. This is set up very much like a cerebellum. And these are like the Purkinje cells of the cerebellum. So it would be as if you had a theory of the cerebellum that didn't include the Purkinje cells. Okay, so what actually happens is that these MG cells inhibit the output cells, and the bulk of the cancellation actually is carried by these guys. So there's a very strong cancellation signal here. I've drawn it as the same sign because there's a flip due to the inhibition. And so it's really the sum of this plus this that cancel that unwanted signal, leaving ideally just the bug. So that's how the system works, okay? The bug plus self-generated signal comes in. This self-generated signal is predicted by plasticity on these synapses, and the result is just a flat signal. Now, what I'll show you later in the talk is that what I said is true. The bulk of the cancellation comes from MG cells, and we think of this as just a little bit of single cell tuning of the thing. So I'm going to talk about this side, but I just want to mention one thing here. The plasticity on this system, there is plasticity here, so what I told you is true, and it's really driven by the error signal being a response. In other words, this plasticity tries to make this response as flat as possible. Of course, it can't predict the bug, so it can't make it totally flat, which is good because that's the point. But all of these predictable signals coming from the self-generated signal are removed because the plasticity, the error signal for the plasticity is a deviation of that output from being flat. Okay, all right, so, but now we're going to ignore the output system for a while because the new thing, basically this new work came about because Nate recorded from the MG cells. So MG cells are although they're more numerous, they're more difficult to record from, and very little work had been done on them. And so their role was not known, and in more particular, the way they learn the way they pull this off was not known. Okay, so I'm going to focus on this part of the system. I'm going to simplify things a little bit. One is, what I'm interested in is how does this system generate, a prediction of this? And so I'm going to get rid of the bug for a while. We don't need the bug because we're just trying to do the cancellation part. The bug will come back at the end. And then I just want to neaten up this system a bit. And now I got to include something that I've left out. And that is the MG cells have got to know about this signal. If they're going to cancel it, they've got to know about it. And they, in fact, these MG cells get this signal through an inhibitory synapse. That's what I've drawn here. Okay. And as I said, the story is going to be that there's plasticity on these synapses, and that, in the end, is going to generate an output there and drive basically a copy of this, but it's not a copy. It's a learned copy. One thing you got to get straight, you could say, well, if you just wanted this thing to come in and go out, that would be very simple. This is not this signal, because if it was, it would also subtract the bug. This is a prediction of this signal generated through that pathway. Okay, so what I have to do is explain how this is done. And yeah.
So they get the signal. Do they get the efference copy, or do they get the actual measured, the red guy that you feed in there?
They get the sensory input.
They get the sensory
Sensory input, yeah, sensory input, and they get this copy from here. Yeah, but through this pathway, yes, just the sensory input. Okay, so the question is, what drives this plasticity? And I'll proceed to explain that. Okay, so the key to this system is that, like in Purkinje cells, and actually like in a lot of cells, there are two kinds of spikes. What I've been talking about are narrow spikes, which are just the regular action potentials that we all know and love. And those are the primary carrier of this signal. Action potentials go down this axon. That's what inhibits the output cells. But there is a second kind of spike called broad spikes. These would be called complex and simple spikes in Purkinje cells. They would be called plateau potentials probably, and spikes in a cortical cell or a hippocampal cell. At any rate, there are these two classes of things. And the broad spikes are broad. That's, and if you measure them from the soma, and this is actually kind of important, the axonal spikes are quite attenuated from at the soma, but these guys which come about in the dendrite are much less attenuated. So from the soma, what you really see is huge broad spikes and broad spikes. Okay, now the key here is that the broad spikes are what govern the plasticity. Again, like in Purkinje cells and like probably, at least somewhat probably in hippocampal and cortical cells. It's these broad spikes that drive the plasticity. And so what I'm going to start to do is forget about the narrow spikes for a second and just describe how do the broad spikes drive this plasticity in order to derive a prediction from here of this shape. Okay, so measured quite a long time ago. In fact, I think this was maybe the first spike timing dependent plasticity that was actually measured again from Curtis Bell's lab. The properties of this plasticity were worked out in a slice preparation, and it goes like this. So if you have a presynaptic spike, meaning let's say that granule cell fires, and it's followed by nothing, by not having a broad spike. So all that happens is this guy fires, the MG cell does not have a broad spike. Then what happens is the synapse gets stronger. So if we're talking about the black MG cell, that synapse would get stronger under that thing. And if you think about, I want to do this to it. On the other hand, if this granule cell has a spike and it is followed by a broad spike within 10 milliseconds, 20 milliseconds, that has the opposite effect. That weakens the synapse. So in this case, this synapse will be weakened. Okay, now if you think about the rate at which these two things are going to occur, this first one just requires a presynaptic spike, so it's going to occur at a rate which is proportional to the rate of granule cell spiking, because that's all you need. Whereas this other process is going to require a coincidence of a granule cell spike and a broad spike. And so it's going to go at a rate proportional to the product of those two rates. And this system will come to equilibrium. So now we have the synapse going up, going down. Where will it come to equilibrium and sit at a constant value? It's going to come in when these two things are proportional to each other, right? When the rate of strengthening is proportional to the rate of weakening. And that obviously occurs when the broad spikes are at a constant rate. So this mechanism has a very nice and very simple consequence. What happens in MG cells is that these synapses will adjust themselves to make the broad spikes at a fixed rate. And that fixed rate is a few hertz. So each of these cells has a set point, and these synapses will twiddle up and down in an effort to make sure this is true. All right? So then how does this solve the problem? Let me go through. Let's imagine initially these synapses are set up so that there's just a constant input coming from all this stuff every time there's a EOD, electric organ discharge. And in that case, there's nothing to cancel this inhibition. So this is an inhibitory input. It's going to inhibit the broad spikes. So the broad spike rate is going to be something like an inverse of that pattern. So that does not make this rule happy, right? This rule is looking for a constant thing. So what's going to happen here? Well, what we might imagine is at the tail end of this, the system is actually at equilibrium. So at this point, it's firing at the appropriate rate. But obviously, if you go, and that means there won't be any change to these late inputs. So a granule cell, let's say firing here at the end, is going to have no plasticity, and this part of the signal won't change. On the other hand, if you look here, this firing rate is higher than that equilibrium, and higher than that equilibrium means you're going to weaken the synapse. And so granule cells that, let's say, fire in this range, in the middle here, are going to be weakened. Those synapses are going to be weakened. And likewise, if we go to this point, that rate is too low. Too low means you strengthen broad spike rate. And so these guys, these early guys in the sequence are going to get strengthened. And so what's going to happen? What's going to happen is you're going to generate the inverse here, right? You're going to generate, what I'm showing you here is what comes in through all these synapses into this black neuron through the granule cell inputs. And gradually, it's going to flip it around so that this guy is at equilibrium. It's satisfying this equation. And now you've extracted a signal here that exactly cancels this, right? And in fact, this process is called cancellation. It just means that you've done what you need to do. This is the predictable part of the sensory signal, and you've generated a copy of it from the corollary discharge pathway. So that's how you solve the problem. Very simple. That's been known for a long time. Okay, now we come to the narrow spikes, and now we've got a dilemma. Because cancellation means these two inputs cancel each other. Well, if these two inputs cancel each other, the narrow spikes should just be flat. And that means there's nothing going to the output cell. That would mean these MJ cells are completely useless. They're not sending any information at all. And that's an illustration of the problem I started with, right? Namely, the signal here that minimizes the error is a constant signal. And that is not a good signal to send to the output. What you want to send to the output is this, right? You want to say, I think a good thing to subtract would be this. So this is why this system has exactly that problem, that the best signal for learning is not the best signal to transmit. So how does it get out of this dilemma? So the way it gets out of this dilemma has to do with this input. It turns out that this input is only involved in the learning. And I will explain biophysically how that happens. But so this, as far as the learning goes, I told you the full story. I told you the truth. But as far as the narrow spike response, it's as if that input is not there. It doesn't do anything. And then you can see you've solved your problem, because then this signal is going to come down completely unimpeded, and it's going to go out in the output, and we've got the answer we want. So the magic here is in this kind of input being a learning input. What on earth does that mean? I'll explain to you. But basically, when we did this project, we realized this is the only way to explain this system. This synapse had to be active during learning and somehow not affect the narrow spikes, and we could explain everything. That bothered Solomon enough, so he actually made a biophysical model, and I think we have a good idea of how this works now. So that's the next step in the process of explaining, what does it mean to have an input that only governs learning. Okay, so let me just say the answer. The answer is that this input is computed by the broad spikes and then transmitted by the narrow spikes. And that's how you get out of this dilemma, that in this computation, that involved completely canceling the broad spike response, but the narrow spikes nevertheless get to transmit the answer. Okay. So what on earth does it mean to have a learning input? So let me go back for a second. Basically, what Solomon did, because we were worried about this, is to build a multi-compartment model from a fill of one of these MG cells. So Nate did a fill. He constructed this model. He put conductances in it, sort of the minimal set that would generate narrow spikes and also broad spikes on the dendrites. He ran the model, I'll show you the answer in a second. But basically, he built this model. I'm always very wary of these models because they involve, potentially tons of parameters. It's a little scary building a multi-compartment model, but he went and did it. I'll give him credit for that. As you can see, it kind of looks like the data. So these would be somatic recordings from a real cell and from a model cell. Here's a broad spike, and it looks pretty good. And then he just tried it. And I guess I should mention, narrow spikes are formed on this axon, which is the red part here, broad spikes on the dendrites. The granulin cell input's going to come in up here. Here's the sensory input. It sort of comes on the lower part, the proximal part of this apical dendrite. So that's the model. So he just tried it. He just built the model and tried it. And here's an example. So he put in this inhibition. He put in a pulse here, and he looked at the broad spike rate. So first he adjusted the cell so spontaneously it would fire 4 hertz broad spikes. That's about right. And then he put in a pulse of inhibition, and no big surprise, the broad spike rate went down. All right, so it means inhibition works. But to our amazement, when that happened, there's absolutely no effect on the narrow spikes whatsoever. It's just, the narrow spike rate, which is adjusted again, it's much higher. It's 50 or 60 hertz in the real cell. So he adjusted the narrow spike rate to be up high. It doesn't even notice the inhibition. Now you do the cancellation. All right, remember in cancellation what you do is you add an amount of granule cell input to exactly cancel this. So you can see he did that by hand. He didn't bother with the learning rule, but he just did it by hand. And you can see in the presence of this inhibition, the broad spikes are constant. And you could imagine that this 4 hertz is that equilibrium where learning would take you. And sure enough, out comes the image of the input in the narrow spike. So this is a case where building the model was trivial. Then we had to figure out how the model worked, which is practically as hard as figuring out how the real cell worked. But anyway, so it worked. So there's nothing magical here. It just worked. Okay, so how does it work? The first observation, which was actually known in the data, but nobody made a big deal of it, is that if you notice, there's always a narrow spike, both in the model and the data, right before a broad spike. Okay? And in the past, it was said, well, whatever depolarization is making the broad spike, it happened to also make a narrow spike. But we don't think that now. We actually think that this narrow spike triggers the broad spike. That is definitely true in the model. And if you look in the data, first of all, it's always true that a narrow spike precedes a broad spike. But in addition, if you look at the EPSPs, they're a tiny fraction of the narrow spike height. The narrow spikes are about 10 millivolts. EPSPs are 1 or 2 millivolts. So basically, they're the only thing big enough to generate a broad spike. And so that's the first observation. And it's basically that you make a narrow spike, it back propagates, it gets highly attenuated. I showed you it's even attenuated in the soma. It back propagates here, and every once in a while it generates a broad spike. Now, every once in a while it's pretty rare, right? Because I said, broad spikes here were going at 4 hertz, narrow spikes at 60 hertz. So this is a pretty unlikely event. And what you see is that the attenuation, the fact that they're attenuated in the soma, that was actually important. Because what's happening is that the narrow spikes are getting attenuated, and they just barely have enough oomph to generate a broad spike. In fact, only 1 in 1 in 20 or something like that can do it. So this is sort of a marginal system, and that's important because it means a very tiny amount of inhibition can kill it, can change it. And that's the key to this system. So let me just think. Yeah, so the way I want you to think of this then is that narrow spikes are occurring all the time, and there's a certain low probability that one of them will just happen to be big enough due to riding on a fluctuation or something to generate a broad spike. So the broad spike rate is going to be some probability times the narrow spike rate. Remember that. Meanwhile, I got to show you a movie that Solomon made. Okay, so in this movie, what you're going to see is first an action potential gets generated in an axon, and then you're going to see that this dendrite gets lit up, and that propagates, and you'll see a broad spike being made. So here's how the broad spike being made. So if you watch here, there's the action potential. It's actually kind of slow, but there it goes, now it's spread. There's the broad spike. Okay? I'll show it to you again, and you'll see what these colors are. But that's the show. Okay, Solomon says I got to show it to you twice. So I got to show it to you twice. Okay, here's the second one. What you're seeing here are the gray is the action potentials in the axon, and there happens to be a second one here. This second one doesn't do anything, so it doesn't matter, but there is another one. And then you're seeing the soma. This is the soma, what it looks like in the soma. Branch 1 is the purple here. Branch 10 is this guy, turquoise or whatever it is. So let me just play the movie again. So the action potential fires. Whoops. And then it takes quite a while actually for the thing to go off, but boom, there's the broad spike. Okay, I just gotta wait for this to be done. All right, so here's how this thing works. Originally, there's in this case, sort of 60 hertz narrow spike rate. Those narrow spikes come back, and with a low probability, as I mentioned, they generate a broad spike. And so if you multiply this by this, you get the broad spike rate, which is low. Okay. Now you turn on the inhibition. As I showed you, the inhibition is too weak and too distal to really have any effect on the narrow spikes, the rate of narrow spikes. But what it does is in the area right where the broad spikes are being made, it pulls these down. The effect of the inhibition is to hyperpolarize that area of the neuron, pulls these down, and now the probability goes extremely low. It's very hard for the system to generate an action potential. And that sets off the learning mechanism, because now the product of this and this is below the target rate. So now those synapses are going to start changing, and they'll start changing in such a way that the granule cell input will push this back up again. Okay? And if you notice, this is actually what happens in the thing. The peak gets back where it was. So those high action potentials can still generate broad spikes. But there's an attenuation of the narrow spike that does not go away. And that's because the inhibition is still there. Remember, the cancellation is just an excitation that has an effect of canceling, but the inhibition's still there, and so the shunting from the inhibition still shortens the action potential. And the result of that is that the probability of generating a spike does not come back to where it was. This little spike is just not as efficient as generating an action potential as that one. But we know that the broad spike rate, if learning is done, has to come back to its original 4 hertz, and the difference is made up by the fact that the narrow spikes have now a higher rate. So that's the transmission of the negative image. It has to do with the fact that the system does not go back to where it started. It gets as close as it can, but the leftover slop is the transmitted negative image. So that's how the system works. Okay, so now I've really shortchanged the experiments, and it's a beautiful work from Nate and his lab. And so let me tell you about the experiments before I wind up. Okay, so from this, what I've told you, if you believe me, we have some predictions. One is that the narrow spike output of these MG cells should match the sensory input to the output cells, because that's the cancellation. It doesn't have to totally match it in amplitude because we have a little slop left over, but these two shapes should match pretty well. Otherwise, it makes no sense what I've been saying. And so you can measure those experimentally, and hold on to your hats, because this involves clever experimentalization. It doesn't involve, optogenetics and expressing genes and all that. It's just good old-fashioned cleverness, and cleverness is hard to explain. But so let's try with the cleverness. Okay, first, this is the original setup that I showed you. This is how the system works, but I misled you in one thing, and that is in these preparations, in the experiments, the fish are actually paralyzed so that you can stick an electrode in their head, and when they're paralyzed, because the electric organ is a muscle, the electric organ actually doesn't work, like all the rest of their muscles. And so this thing that they generate is not there. Now, they keep generating commands. maybe I should use this one. This one's kind of pooping out. They still generate commands, so they're trying to generate it, but this does not happen. And what happens is in the lab, you replace it by a fake. You generate your own electric field that matches what the fish is expecting, and you tie it to the command. So you measure from the nerve bundle that sends back the signal to the tail. If you see a command, you trigger the electric field in the lab, boom, you get an electric field, and the fish is totally fine. Thinks everything's working. Okay, so that's how it goes. But because of that, you can play all sorts of tricks. So one of the tricks you can play is you can generate this electric field without a command. So just generate it at the wrong time. And when you do that, the fish cannot generate these compensating signals because it doesn't know the electric field's about to be pulsed. And that allows you on the output now to measure the sensory signal without all of this cancellation going on. So you can measure the sensory signal. That's trick one. So measure the response that you're supposed to be canceling. Now, that's enough. That gets us there as far as this prediction, because this can be measured just by recording from an MG cell. It's just the MG cell output, no big deal. This can now be recorded by the trick I just showed you, and you can see if they match. And here's the match. Actually, about 10 or so MG cells converge onto each output cell, so that this is the sum of 10, or the average of 10. And you know, it works pretty well. So the shape matches quite well. In the black is the sensory response that has to be canceled from one of the output cells, and here in green is the MG cell summed response, and remember that gets flipped by inhibition. Okay, the prediction worked. It's a pretty good match. Okay, so now the other thing that I told you is that this one, as I've drawn it, this negative image is much bigger than this one. Of course, it's flipped through the synapse. And so we also have experimental evidence that that's true, the royal we here. And that's obtained in another way. It's doubly tricky. So the other thing you can do to trick the fish is it generates a command, but you don't turn on the electric field. You leave it off. You can't, if you do this for too long, the fish will relearn that there's no electric field. But if you do it a few times, the fish will not compensate. So that removes the sensory pathway. There's nothing there anymore. And that allows you to measure the sum of these two guys, right? So you could measure the total cancellation signal at the output. That's the sum of these two. But obviously, if you want to compare these two to each other, you've got to measure one of them separately. And that's done very cleverly. So in this case, we're back to the regular setup. But what Nate did in this case was to inject into the cell, triggered by the command, the inverse of the sensory signal. So in other words, Nate himself did the cancellation in this output cell. Right? That means that the plasticity at these synapses has no error signal, right? Everything, as far as that output's concerned, everything's been canceled. And that eliminates this self-generated negative image. But of course, the MG cells don't know that Nate's doing this trick, and so they happily make their signal. And so now on the output, when you restore the system, you can measure just this signal by itself. Okay, that's the tricks. You can wake up now if you're a theorist. All right, so here's the result of that. Do I have another? Yeah. So the black is the total negative image signal. The green is what happens when you trick the output cell into not doing anything. And so this little bit is the remaining. So you can see that it's true what I said. The majority of the cancellation signal is actually coming from the MG cells. Okay. All right, so I'm actually winding down, so we'll have time for questions, I guess. All right, so there are two things I didn't tell you about this system, and then I've really kind of, I think, told you more or less the truth. One is an aggravating thing that I've ignored, and that is that the sensory input actually comes through the pathway I've talked about, but through another pathway. Yeah, you have a question? Yeah. So within about 5 minutes, you see it getting there, and it takes about an hour to really accomplish a nice cancellation. So that's the time scale. Okay, so I'll just say that, you know, the reason that this occurs at all is because the properties of the water seasonally change. So the electrical properties of the water where the fish lives in this muddy water change. So it has to do it during the course of the year. Also, of course, the fish grows. The fish may get injured and you know, change the receptor pattern on its skin. So this is an ongoing plasticity through the life of the fish, but it doesn't have to be, you know, within milliseconds. Waiting for an hour is fine. It's fast enough. So that's the time scale. Okay, so what I was telling you is that there's actually sensory input that affects the narrow spikes, which I didn't account for. And that's because it just gets canceled by this system, and we have no idea why it's there, to tell you the truth. So, but it's there. It doesn't change the story I told you at all. It just makes it more difficult to see what's going on, and it drove us crazy. But I didn't drive you crazy with it. The other thing is that I've only described half the system, but it's easy. Don't worry, I don't have to give my whole talk again. And that is, like many systems that have you know, on side and an off side, this system is duplicated by another set of cells that do exactly the opposite of everything I said. So here, these are called output E cells because they're excited by the stimulus. Correspondingly, there's a set of output I cells that are inhibited by the synapse, exactly backwards. So their plasticity is driven in the opposite direction, and their MG cells are rather than being inhibited to learn, are disinhibited to learn. Everything's backwards. And so they learn a backwards signal. Okay, but that's the end of the story. But and that means there are two kinds of MG cells. We call these BS minus. They're inhibited. Their broad spikes are inhibited by the stimulus. And BS plus. Their broad spikes are essentially excited by this stimulus because they're disinhibited. Okay, but this actually was a scary moment, because what you can see is that if what I'm telling you is going to work, you have to have it that the BS minus cells are inhibiting the output E cells. If they inhibited these guys, it would be the backward sign. And it must be that the BS plus cells are inhibiting the output I cells. Again, otherwise the whole thing wouldn't work. And you might think this should be obvious. The reason it's not obvious is because of these inputs, which confuse the definition of the BS minus and BS plus. So in the past, it was realized that there were two kinds of cells, but it wasn't clear what the basis. There was anatomical basis for two cells, but it wasn't clear what the functional basis were. So this is an absolute prediction of the model. This is, if you're a modeler, you try to avoid moments like this where an experimentalist can completely kill you. But Nate had to find this, or otherwise we were dead, and Solomon would have to go back and do a different thesis project. So Nate did the experiments. What you have to do, what he had to do, is to measure these cells and determine functionally whether they're BS minus or BS plus, then fill them. And then the good news is that this is a layered system with the output E and the output I cells receiving their input in different layers. That's what all this shows. I'm not going to go into detail, but the dendrites of the output E and I cells are in different layers of the structure. So you have to trace these axons and see which layer do they go to, and the answer is it works. So the BS minus cells that go to the output E layer, that's this green stuff, and the BS plus cells go to the output I layer. So the anatomy worked. Anatomy to the rescue. Okay. All right, so I'll just end with coming back and saying, all right, all this cancellation, you've blown an hour of your time explaining it. Does this actually matter to the fish? I don't know if it matters to you, but does it even matter to the fish? So at least I can convince you that the fish cares. And that's a very beautiful set of experiments done by Armin in Nate's, a graduate student in Nate Sautel's lab. And it uses the fact that the fish are kind to you. So the poor fish are paralyzed, right? They're not even making their own electric field. You got to fake it for them and all that. But if they, when they're hunting, if they sense a prey in the environment, what they do is they speed up the rate of their electric organ discharge. The reason they do that is because they use the active system as well to find the prey. So they get excited and they accelerate. And even in the paralyzed state, if they sense a prey-like signal, here are the electric organ discharge, the fish will accelerate its electric organs. So in other words, the fish tells you whether it sees the prey or not. And that means you can do an experiment to see when can it see a prey and when can't it see a prey. And here's those experiments. So these experiments started with no mimic. So meaning the electric field that duplicates the missing electric organ discharge field is just turned off. There's no electric field. When you do that, within, again, this hour, the fish will adjust to having no electric field. It's quite amazing. This system, that's something that the fish would never experience in the natural world. But this is a very plastic system, and it can easily adjust to having no field. So it just turns off all those predictions and predicts zero. Okay, and in that case, the fish is quite good at seeing a mimic. So this is the change in command rate when the mimic was turned on. You can see that fish did accelerate its command. But then you turn on the mimic. So the fish has adapted itself to no electric field. Now you suddenly turn it on, and the fish become terrible at seeing the prey. They can't detect it. That's because, as I've been advertising, it's covered up by the big self field. But this is a learning system, so if you wait for an hour or so, the fish can learn to find the mimic again. That's because they subtract out the electric field. And actually, in these experiments, Armin showed that that's what's happening, that the self-generated field is being subtracted, the fish can find the mimic, and then you can trick the fish in a way that it can never recover from, and that is if you put the electric field that duplicates the electric organ discharge, but you disconnect it from the command. So now the fish cannot predict it, and in that case, the fish can never learn to find the prey. So this system really does allow the fish to find the prey. The fish is telling you itself. Okay, so I just want to wind up with some lessons from this system. So first of all, I think that, to my knowledge, this is the first time that learning has been studied in a layered biological system, where you're looking at learning that's back from the effect that you're studying. I may be wrong about that, but at any rate, it is a description of learning in a layered network. And it's an example of this distributed error signal. So the exact same error signal that works here is also at work here, namely, make yourself a flat response. But the dilemma is solved because that flat response is the broad spike response, not the narrow spike response. The other thing is it's a continuous learning system, and that also is involved with this resolving the dilemma between learning signals and signaling signals. So in artificial networks, usually you have a learning phase and an operating phase. And the reason for that is the outputs that drive learning are incompatible with the operating outputs. Here, there's no problem, again, because the broad spikes and narrow spikes are working in parallel and they don't interfere with each other. Okay, so this idea, in this system, you have a resolution of this dilemma between learning and signaling. The other thing that is a lesson of this, that's also come out of Sarah Beller work, actually, by Reza Shadmir, is that if you try to understand the structure of this system, namely this BS minus cells are coupled to output E's and BS plus to output I's, if you looked at the conventional responses, and this has to do with, I guess I left them out here, the way the narrow spikes respond to the stimulus, this wiring would make no sense. You could not figure out why one cell, because you can have MG cells that respond one way, coupled to output E, and then that respond completely the opposite way, output E. And in fact, you know, people before us and we ourselves could make no sense of this wiring pattern from looking the conventional way, which was sort of say, well, maybe cells, you know, with similar responses get excitation. Maybe, you know, these have opposite responses so that they inhibit. It doesn't work. What does work is if you look at the system from the point of view of learning. In other words, this BS is important. It's the broad spike. It's the effect of sensory input on the broad spikes, which means the effect of sensory input on learning, by which you have to sort out this circuit. And I think that's in whatever circuit you study, keep it in the back of your mind, that maybe to understand this circuit, I don't just have to think about how it functions, I have to think about how it learns. All right, I will end there. Thank you.
We have time for questions for Larry.
Yeah.
If I understand correctly, the circuit and the model are for any spatial domain of the sensory
Yeah,
system.
very good, yeah.
I'm wondering, so how does the fish use the somatotopic organization of this rostrocaudal antenna? And is there any cross connection
Yeah.
that gives additional information?
Yeah, exactly right. I should have mentioned this, so I'm glad you brought it up. This is considering sort of a pixel view of it. And very little is known about integration across space. So I would say that's sort of a next problem in this system about how the spatial thing, and in fact, Nate is starting to study that, because one thing he's now recording in freely moving fish. So you can actually watch them approach a prey. Very good question, but very bad answer.
One thing that happened with there's another fish, they have this avoidance.
Yeah,
avoidance
yeah.
response. So, but that seems to happen much faster than 1 hour.
So it's also the other kind of fish. So there are two kinds of electric fish. And that this one chirps, you know, it makes a thing. Those are sinusoidal emitting electric fields. And so, you know, the ones that do the jamming avoidance is a different fish, and it's actually quite a different system. They don't have this system. So yes, it's interesting, and yes, that's very fast, but it's a different beast. Yeah. Yeah, go ahead.
How complex is the waveform of the mimic potential that the fish can learn to answer? Is it like a specific form, or can
Yeah.
it be?
I think it can be quite varied. Where it would get hard is if you, in the initial stage, I think you could make it quite weird looking. For example, you can reverse it and they can deal with that. Later on, if you remember that granule cell the responses, there isn't a whole lot going on late. So I think if you did something too big late, you'd have problems. It would have problems. But you know, this system is just incredibly plastic. There are adjustments within the active system for the position of the tail. As you might imagine, if the tail moves, the electric field changes on one side of the body or the other. So that's also done by a circuit like this. You can actually reverse that and make it stronger when the tail's on the other side. The fish deals with it. So it's a very general learning system.
So when you were like developing this research, I imagine when you first look at like the individual pieces, like the units, who inhibits who, and when you finally put it together, was it like obvious that this is how the circuit looks like? Or was there?
Yeah, so I mean, it was known that MG cells inhibit, and that what wasn't known is this classification. That's the new thing. So the general structure was known. It was known that there were two anatomical types of MG cells, but the map onto function wasn't done. So that's the piece that was added here.
And all the other parts of the circuit, like the VOD, like.
Yeah, Curtis Bell. I mean, Curtis Bell deserves a huge amount of credit for doing a huge amount of this. So we had Curtis Bell. That's the answer. You got to have a Curtis Bell.
So why should there be, you know, both parallel fiber inputs and the MG inputs to the output?
Yeah. So we worked very hard on that, and I don't really have a great answer. We have evidence that this plasticity saturates before it can account for the whole size of this. And this doesn't because, among other things, they're 10 to 1. So it could be that just the dynamic range of this synapse wasn't big enough to handle the problem. I think of this as a tune-up. So remember, this is calculating a negative image on the basis of this input. If this input is a little bit different in some way, maybe it samples from slightly displaced sensors or something, you might need a little tune-up for each cell. So I think of that as a kind of a tuning up.
This in particular is like the MG apple, like more sort of plastic in its shape.
Yeah.
What was your difference in the rate of learning?
Yeah, so no, not apparently. So we have an argument that's based on, again, thing. There are also connections from here to here. So I'll give you one of our arguments. It's just I don't know if it's right. And that is, if you think about the speed of learning, you can increase the broad spike rate a lot. It starts at a few hertz. You can increase it to 10 hertz plus. So in that direction, you can drive fast learning. But you can't decrease it below 0. So you have an asymmetry in the learning. And so we had an idea that one side learns the plus side and one side learns the minus side, and then they share that. Curtis Bell also thought of that idea, actually, and we don't have evidence that it's true, though.
What about between the blue and the black? Between the parallel fiber optics and the
So is this plasticity faster than this? For example, the trouble is, so the tricks of the trade. In the past, when people talked about plasticity here, including Curtis Bell, including us, the plasticity here has never been characterized. They use this plasticity over here, only they use narrow spikes instead of broad spikes. So we don't really, we know that these are plastic because we see this signal, but we don't know in detail the nature of that plasticity.
Isn't it known from the clever experiment where you fed back
Yeah.
the output, as you can see how
Yeah, so these form at about the same rate. You don't see a big difference. It would have been nice if this was fast and this was slow or something like that, but that didn't happen. So I don't totally know why it's a two-stage system. We don't know.
So in this very specific example, like the distributed local error is just each neuron trying to generate like a flat output, right? So this flat output is kind of coupled to the two different spikes.
Mm-hmm.
So I'm wondering, like in terms of general like target or global like targets. So is that, for example, you now trying to learn something else instead of trying to cancel out. Do you think the approach is general?
Yeah, so yeah, what would you do in machine learning with this? Basically, this is like cloning a signal. It allows you to be given a signal and said, from this from this stream here, generate this signal. So it's a cloning device. So you got to figure out if there's a problem that you can solve it that involves cloning.
Do you have any idea when you have the sinus is not shown, but why would the MG cell receive an excitatory input and
This one that I didn't show, yeah, because I hate it. I don't know. I really don't know because it just gets canceled. It doesn't seem to do anything.
it gets the opposite sign on both sides?
Yeah, this one can be either. It can be either, actually. That's why it confused the daylights out of us, because this one can be either excitatory or inhibitory, but this one is always inhibitory. I don't know what it does. Maybe it translates a little bit of the bug signal, but I don't know why it would do that. That one's a mystery.